Hello there. Welcome back to another exciting episode of Teach Me in 10. I'm your host, Lucy Lawrence, and this is Technology Network's video series where we challenge scientists to teach us fascinating research and scientific concepts all in less than 10 minutes. In this episode, I'm speaking with Tom Charlesworth, Director of Market Strategy and Corporate Development at biomodal, and we'll be discussing the power of the six-base genome for transformative insights into current and future states of disease. But before we go any further, I am delighted to tell you that this episode is brought to you by biomodal. Now, biomodal is an omics-based life sciences technology and analytics company delivering products that bring the dynamism of their ever-changing biology into focus. Their groundbreaking multi-omic solution, Duet Evoke, reveals the power of the six-base genome by distinguishing 5-methylcytosine and 5-hydroxymethylcytosine together with all 4 canonical bases to measure multiple modes of biology from a single 5 nanogram DNA sample in a single experiment. To learn more about the Duet multi-omic solution Evoke, you can click the links that we've left in the description for you. But without further ado, you're about to watch this incredible Teach Me in 10 in full. Hi Tom, how are you doing?
I'm very well, thank you, Lucy.
Perfect. So, as we only have 10 minutes on the clock, we best get started. So, firstly, what is a six-base genome?
So the six-base genome is your canonical 4 DNA bases, so what we might call the 4-base genome. You've got your A, your T, your G, and your C, but also layering on 2 additional layers of information. So you've got a 5-methylcytosine and a 5-hydroxymethylcytosine. So traditionally, you'd have to do a couple of different workflows to get that additional MC and HMC, the sort of 5th and 6th bases of the genome. Whereas with that, we have a solution that provides all of those 5th and 6th bases with your full 4-base sort of genetic genome in one workflow. The idea being getting complete genetic data and epigenetic data from a single experiment in a single data set. And so that is your 6-base genome.
Fantastic. And then from that, what type of insights can be derived from the 6-base genome, and how do the methylated bases 5MC and 5HMC contribute?
Sure. So I think I'm going to focus on talking about liquid biopsy as an application of the 6-base genome. So liquid biopsy is where you have a cell-free DNA sample and you are trying to use that to understand something about someone's disease state. So understanding, predicting or early detection of cancer is the most common use case for liquid biopsy. In that instance, the 6-base genome provides you with a true multi-omic data set, if you like, from which you can derive a sort of broad range of features that you could use to build into a classifier to perhaps detect cancer earlier or detect disease recurrence or get some insight into the biology of the cancer that an individual might have, and that might guide treatment decisions or other sort of clinical decision making. As far as so when I talk about multi-omic data that you can get from the 6-base genome, obviously you have your genetic data, so your SNPs, your CNVs, so sort of point mutations and more sort of growth aberrations in the genome. You also have the epigenetic bases, the methylcytosine and hydroxy. And then you can start to look into sort of other features that you might want to get from your cell-free DNA sample, such as some sort of fragment information. And we've got some product sheets and posters that evidence how you can get that sort of multi-omic data from your single sample using the 6-base genome. But to take that further, those are kind of, I guess, your vanilla features that you might want to use. But with the addition of MC and HMC, you can also start to look within a cell-free DNA sample at other sort of regulatory regions of the genome, for example. So we have some data where we've looked in a cohort of 88 cell-free DNA samples from people that are either healthy or they've got stage 1, 2, 3, or 4 colorectal cancer. And in that cohort, we've built a classifier for detecting stage 4 cancer, differentiating that from healthy. Not a very difficult problem to try and solve, but we found features. Some of the features that were most discriminatory were enhancers, which are known to be relevant to the biology of MC and HMC. And if you take an enhancer that discriminates between stage 4 and healthy, and you look back in the earlier stages of disease, because you have that 2-dimensional information, you can tease apart the differences between a healthy individual and an early stage cancer sort of patient in a way that you wouldn't be able to do without the addition of the MC and the HMC, the sort of fifth and sixth bases of the genome. And yeah, we've got some data that sort of demonstrates that, and you're tracking a repressed state of an enhancer, MC-HMC, where you've got a bit of a mix, you've got a primed enhancer, an area that's about to be switched on, and then a repressed enhancer. So you're getting an indication as to, within a cell-free DNA sample, firstly, you're getting an entirely new feature you couldn't otherwise get, but you're also getting, in that in-between state, that prime state, where you have a mixture of MC and HMC, you're getting some information that tells you something about what might be happening in the future, because you know that region is about to be activated, even if it isn't active when you're getting that sort of snapshot of information.
What does the workflow entail to generate this level of data and insight?
Sure. So it's a single workflow. It's composed of an assay that a sort of wet lab assay. So you take your DNA sample, you run it through our assay to create a sequencing-ready library that you would load onto your standard Illumina sequencer. It's compatible with very low input DNA. So in the liquid biopsy example, you can go as low as 5 milligrams of cell-free DNA. You then you have your FASTQ that comes off the sequencer, and you can push that through our pipeline, the Duet pipeline, and that does what we call read resolution. It does quality filtering, trimming, and all the rest of it, and provides you with what we call the resolved genome, the 6-base genome, and a number of sort of standard file formats. Now, the kind of core concept of the workflow is that you have an original fragment to which you append a hairpin, you have a copy step, and then there's some differential sort of enzymatic steps. There's methyl transferase, there's deamination and protection steps that gives you a different context across the CpG and across the DNA duplex that allows you to derive either an unmodified cytosine, a methyl cytosine, or a hydroxymethyl cytosine. And it's that logic, that sort of molecular biological logic, that is resolved in the pipeline that you would use to process your sample and produce the 6-base genome.
Fantastic. And what additional predictive models can be generated from this 6-base genome data?
Sure. So I've talked a bit about looking at enhancer status, and that sort of speaking to, you can get that in-between state of enhancers where they're not off, they're not on, but they are about to be on, because there is a demethylation pathway where you go from MC to HMC and then demethylation, and that follows an activation trajectory. So that's one piece, and you can apply that in a liquid biopsy setting, but also you can start to look at that in a sort of a genomic DNA setting as well. But we also have done some work internally on deep sequenced mouse E14 stem cells, where you have orthogonal measurements. So you have gene expression measurements from RNA-seq experiments, and you also have ATAC-seq experimental data that gives you chromatin accessibility. And we've used the MC and HMC features across a couple of different sort of genomic regions to build fairly simple classifiers that demonstrate that with the 6-base genome, you are able to fairly accurately predict levels of gene expression and predict levels of chromatin accessibility from the 6-base genome as a kind of your baseline. So you're getting the 6-base genome with all the features I talked about previously in the liquid biopsy setting, which you can also apply to your genomic DNA setting as well. But you're also, what the models are sort of demonstrating the promise of is that you can start to get some more functional readouts that go beyond simply additional features. You're starting to say, well, where there are differences in MC and HMC, I can get some idea of predicting what's happening from a gene expression perspective, from a chromatin accessibility perspective, and then you can combine that with information you might get from some of those enhancer kind of measurements about predicting where the biology of your cell might be going in future as well.
Fantastic. And then finally, as we're almost out of time, but what resources are available for viewers to learn a little bit more?
If you go to our website, we have the product is called the Duet Multiomic Solution Folk. We have the product page with some of this sort of outlined up just straight up on the website, but we also have a number of resources that you can get hold of differing levels of complexity. So you can take a look at the product sheet where a lot of this data is included. We have an application note that covers the CRC cell-free DNA example that I spoke about and goes into some detail about the features that you can derive from that data set. And then we have an application note from one of our early access customers, liquid biopsy application, again, looking at hepatocellular carcinoma as a kind of use case. And if you really want to, there are also the world's first 6-base genome is publicly available. We've deposited in the Geo repository, and you can again follow a link from our website to get hold of that if you really want to dig.
Wonderful. Well, thank you so much for your time, Tom. It's been an absolute pleasure speaking with you.
Thanks again. It's been great to talk about the products and what you can do with the 6-base genome.
Thanks so much for watching this Teach Me in 10, and we hope we see you again very soon.